investigating enzyme action, the effect of temperature on catalase activity. Enzymes are special proteins that act as biological catalysts, speeding up chemical reactions in living organisms. One of these enzymes is catalase, commonly found in many plant and animal tissues such as potatoes. Catalase plays a vital role in breaking down hydrogen peroxide, a toxic byproduct of cellular metabolism, into harmless water and oxygen. In this experiment, we aim to investigate how temperature affects the activity of catalase by observing the height of foam produced during the reaction between hydrogen peroxide and the catalase enzyme extracted from potato. Fresh potato serves as the natural source of catalase enzyme. Hydrogen peroxide, 3%, acts as the substrate. 4 test tubes and a test tube rack to hold and organize the samples. Containers or water baths set at different temperatures, 0 degrees Celsius, 25 degrees Celsius, 37 degrees Celsius, and 100 degrees Celsius. Food thermometer to monitor water bath temperatures. Measuring ruler to measure the foam height. Knife and cutting board to cut equal potato pieces. Safety goggles and gloves for laboratory safety. First, we cut the potato into equal small pieces, each approximately 1 cm cube to ensure consistent enzyme amounts. We prepared 4 baths or containers representing different temperatures conditions. 0 degrees Celsius ice bath, 25 degrees Celsius room temperature, 37 degrees Celsius warm water bath, and 100 degrees Celsius boiling water bath. We verified each temperatures using a food thermometer. Four test tubes were labeled 1 to 4, placed on a test tube rack, and filled with equal pieces of potato. Each test tube was placed in its corresponding water bath for 10 to 15 minutes to allow the enzyme to adjust to its environment. After equilibration, we added equal volumes of hydrogen peroxide to each test tube. We initially planned to use a pipette, but it did not dispense properly, so we poured the hydrogen peroxide carefully until it reached the height of the potato piece. We started the timer immediately after adding the hydrogen peroxide and allowed the reaction to proceed for 30 seconds. After 30 seconds, we measured the foam height in each test tube using a ruler. The foam height represents the oxygen released at catalase breakdowns hydrogen peroxide. Based on our results, enzyme activity appeared lowest at 0 degrees Celsius, showing no visible reaction. As the temperature increased to 25 degrees Celsius and 37 degrees Celsius, the foam height slightly increased, indicating higher enzyme activity. The boiling water bath, 100 degrees Celsius, produced the most visible foam, measuring about 0.3 centimeter. From our observations, we can conclude that temperature significantly affects catalase enzyme activity. As temperature increases, the rate of reaction also increases, resulting in the higher foam formation. However, it's important to note that at extremely high temperatures, enzymes usually become denatured, meaning they lost their shape and can no longer function properly. In our case, while the boiling temperature showed foam, it may not entirely be due to enzyme activity, but rather the physical effects of heat causing bubbling. Therefore, catalase is expected to work best at moderate temperatures, similar to body temperature around 37 degrees Celsius. Catalase breaks down hydrogen peroxide into water and oxygen. Temperature directly influences enzyme activity. The reaction rate increases with temperature up to a certain point, after which extreme heat can denature the enzyme. This experiment highlights the importance of maintaining optimal temperature conditions for enzymes to function effectively, just like in living organisms.
At yan ang epekto ng temperatura sa enzyme na catalase. Tandaan, maging ligtas sa bawat eksperimento at patuloy na tuklasin ang agham.